So we are pleased to welcome Dr. Elizabeth Riley, who received her undergraduate degree in biological engineering from MIT and her PhD in neuroscience from Boston University School of Medicine. She then trained in human neuroscience at the Boston VA and Cornell University. Dr. Riley combines a technical and mechanistic orientation with a broad interest in human health across the lifespan and across and within cultures. Her primary focus is in developing and refining the understanding of modulatory neurotransmitter systems in the brain, especially the locus coeruleus norepinephrine system. Clearly, I'm not a neuroscientist, including its relationships with neurodegenerative disease, somatic health, and psychosocial health. She uses MRI, pupillometry, and vagus nerve stimulation as tools to assess the state of noradrenergic system. In all her work, she strives to generate and extend a sense of community to her colleagues, students, research participants, and the greater world through genuine love of knowledge, ethical leadership, cultural competence, and dedication to diversity.
Oh, how's the sound? It's okay? All right. Thank you everybody for being here today. And I'm sorry about all the $10 words. You all know how to pronounce them after this hour. So we're going to talk about finding the earliest beginnings of Alzheimer's disease. And this little blue speck with an arrow is an in-joke. You'll know what it means by the end, okay? We're going to come back to it. So who is this talk for? Of course, it's for you and whoever's listening on Zoom. It's for anybody who's interested in cognitive health across the lifespan. Alzheimer's disease and other neurodegenerative diseases disproportionately affect certain groups. Right now, those groups include women, Black and Hispanic people, certain health conditions, and certain genotypes. We work in partnership with communities to build a program that represents the full reality of all the types of people and all the types of risk factors that impact this disease. And that's not just fair, it's also necessary for accurate science. Just want to get that said. So let's be detectives. I picked my favorites, but now it's all of you as well. I want you to get into a detective mindset, and I'm going to ask a few questions that I'm not intending for people to know the right answers to, but if you call something out, I'll repeat what you said so that people online can hear. I think I'm going to have to go with Mulder and Scully as my ultimate favorites, but you can pick your own. Okay, so if we want to figure out what causes Alzheimer's disease and stop it, where should we look? And before you answer, pretend it's 3,000 years ago. Just for a minute. Where would you look if you were living 3,000 years ago? Remember what they knew then. What do you think? Anybody? The heart. Yep, that's a great answer. I think that is where people considered thinking to come from a lot of the time back then. Anybody else? Witchcraft, sins, ate the wrong stuff, bad air. A lot of that kind of thing could be true. I think I'm going to click OK so that that doesn't cover the top of the screen. There is a whole world of history, of health history, and I know very little about it. But in the reading I've done, we find that about 2,000 years ago, people had very little sense of connection between the brain itself, which they often thought was sort of fluffy stuffing in your head, and diseases of the brain. Now let's move forward 1,000 years. What do you think where would people look for Alzheimer's disease then? What would they think it was caused by? Any guesses? Old age. Sure, yeah, maybe more people are living to be old. Maybe there's more of a sense of what medicine of old age looks like. Maybe they're just saying it's just something that happens when you get old. Any other ideas? All right. Around this time, there were doctors, Arabic and Persian doctors, who were making the connections between pathology of the brain, things like head injuries, brains that look different, and neurological symptoms and problems. And they're often referred to as the fathers of modern medicine, especially the person who's called Avicenna in English. I can't pronounce the Persian name properly. And then, does anybody know when the famous German physician Alzheimer created the diagnosis of Alzheimer's disease? It was 1906. So at that time, he had a patient, August Dieter, who was only 51 years old and had a very rapid onset and early onset Alzheimer's disease. And of course, he named the disease after himself, or rather, actually, I think a colleague of his named it after him after he wrote a paper describing it. So by that point, people kind of had a sense that this had something to do with the head. So that's what they saw. This is what Alzheimer himself saw. He looked at her brain and he saw that something was very wrong with the brain. It was quite shrunken. It looked dark. A lot of the mass had been lost. But why? Why does it look that way? What happened? This is a good detective question. What would you guess? Like, what went wrong with this brain? Yeah, no kidding. Yeah, definitely. Okay, didn't get enough blood. That's a great idea. Like, it withered away because the blood supply was cut off. That's something that's analogous to other physical problems that you might have. So people might think like, okay, it wasn't sufficiently supplied by the blood. Does anybody have any other guesses of what a detective of this era might think went wrong with this brain? Sure, yeah, head injury. Maybe this is the consequence of an injury that wasn't recovered from. Yeah.
Poisons in the air.
Yep, maybe the brain was very sensitive to some kind of poison. Okay, so Alzheimer himself took a microscope and had a look inside this brain and was able to see that it had two major problems. It had plaques and tangles. Plaques are gunk that accumulates outside the cells in the brain, and tangles are gunk that accumulates inside the cells in the brain. And the difference between the plaques and the tangles is just what stuff that gunk was made of. So the plaques were made of amyloid beta proteins, which are variously referred to as A beta. And also the tangles were made of phospho-tau, sometimes just tau, p-tau. It all means the same thing. And the brain was full of them. The cells were kind of choked up with tangles inside, and the outside of the cell was covered in plaque. And the brain had shrunk a lot. So what would we think to do about a brain that's full of plaques and tangles? I mean, it says it here, but it's a pretty obvious thing for a detective to think, like, this brain is full of plaques. Let's get them out. Well, that took a long time. It took about 115 years. And in fact, it was only achieved properly 2 years ago. And so a drug was developed, 3 drugs now, actually, lecanemab, donanemab, and aducanumab. They were approved. And I want to show you what this graph tells us. This is time, more than 50 weeks. So it's a long time, more than a year. And this is how much amyloid plaque is in the brain. In the gray line is the control group that didn't get the medicine. So as time went on, they didn't have any less amyloid plaque in their brain. In fact, they had a little bit more. And this is the group that was treated with lecanemab, the new drug. And what does it do to the plaques in the brain? Yeah, it reduces it, like a lot. This is about a 60% reduction. So more than half of the plaque in the brain was removed by this medicine successfully. Amazing. But what happened to these people? This is a different graph of more than a year. So this is in months. So we have a 1.5 year time of taking this medicine. And this axis shows how well people were doing on their cognitive tests relative to how well they were doing right at the start. So at 0, they haven't gotten any better or worse. Over time, scores in the green indicate people getting better, having better cognition than they did before. And scores in the orange indicate getting worse. So the trace with the open squares is the drug, and the trace with the black squares is the placebo. So what did the drug do? Anybody put it into easy words? It helped a little bit insofar as people didn't get worse as fast as they did before. Did it make anyone think better? No. That's pretty surprising, I think, for people who have been for more than 100 years aware that the brain fills up with amyloid plaque. Whenever you look at an Alzheimer's brain, they always have amyloid plaque, every time. So why didn't it work? What happened? There's a few problems. The first problem is that amyloid turns out to not be a very specific marker. It's so common with Alzheimer's disease that it's pretty much inevitable. If you have Alzheimer's, you have amyloid plaque. But it turns out you can have plaque and not have Alzheimer's disease. And that's shown by this type of picture here. This is a PET scan. So in a living person, you can take a picture using a fancy PET scanner of how much amyloid plaque there is in the brain to see what's in there. And a normal amyloid negative scan would look something like this. So this is a person without amyloid in the brain. Here's a person with amyloid in the brain, but who doesn't have Alzheimer's disease, meaning they don't have any symptoms of cognitive decline. And here is a person with Alzheimer's disease. So Alzheimer's always has amyloid, but having amyloid doesn't always mean Alzheimer's. So that's not a very good starting point for figuring out how to cure the disease. What's up with this? Well, there were two types of plaque in the brain, right? Two types of buildup. There was the amyloid and the tau. Maybe tau is a better marker. Maybe looking at amyloid isn't a great place to start. So now you can do a tau PET scan too. You can use a scanner to have a look in a living person to see how much tau is in their brain. And it is a little bit better. So this is a person with a healthy person for a scan for amyloid. And here's an Alzheimer's amyloid scan. And just like the difference between these two is not much. The difference between a person with a healthy amount of tau and an Alzheimer's amount of tau is a little bigger. You can tend to see a larger difference, but it's not a perfect marker. And this is where I'm going to share two analogies, neither of which is perfect, but both of them explain, at least to some extent, why removing amyloid didn't cure disease. Taking away the amyloid when a person already has an Alzheimer's diagnosis is kind of like taking a poison ivy leaf out of your sleeve after you already had a horrible rash for 2 weeks. It's maybe a good idea, maybe you should take it out, but it isn't necessarily going to cure your rash. In fact, it wouldn't be reasonable to think that it would cure your rash at all. And another idea with a more common saying is it's not so helpful to shut the barn door after your horse already escaped. So this is probably the most complex figure I'm going to show today, I hope, and I just want to take a minute with it. This is a modern understanding of how Alzheimer's gets started, broadly speaking. And what it's showing us is across time here, a long, long time, probably 20 or 30 years. And then on the vertical axis, on this side, it's showing normal at the bottom and abnormal at the top. And that's for all the lines except for the blue one. You can ignore the blue one just for now. In Alzheimer's disease, we start with A beta deposition. And by the time there's a lot of A beta, we may not even be in the prodromal phase of Alzheimer's disease yet. But after A beta begins to build up, tau begins to build up. And even then, after there's a lot of tau, we may still not be in a disease state yet. After that, neurodegeneration begins. Now, of course, the big question is why? And the brief answer I'm going to give you today is because there is what's called neuroinflammation, meaning your body recruits its immune system in the brain to try to deal with all this gunk that's building up all over the place. And in the process of doing that, it damages your cells, leading to neurodegeneration, the death of cells. And then after that's been going on for a while, only then does cognition begin to change. So if we are looking for a brain that has a lot of amyloid and a lot of tau, both of those markers clearly abnormal, and a person who has maybe some very early signs, we're kind of here in the timeline. But when did it actually start? Way back here. Like this process got going decades earlier. So amyloid isn't a specific enough marker. Removing it doesn't work. Finding amyloid and tau together with brain volume loss is too late. So let's put on our detective hats again. We're probably frustrated detectives going back to the drawing board. I think that's where the field is right now. We've had to go back to the drawing board. Removing amyloid isn't the whole answer. It might be very helpful in early stages. There's not to say there's no good in it at all, but it's not the answer to curing disease. Identifying widespread amyloid and tau is not the whole answer. It's either not specific or it's too late. We know the disease process begins undetected right now. How can we spot it? So here's my proposition. What if we try to figure out where tau tangles first appear? Tau tangles are a more specific marker of Alzheimer's disease than amyloid. So let's leave amyloid aside for now. Let's see if we can figure out what parts of the brain we need to watch very, very closely so that when they begin to have minute changes in function before they're dying, we can pinpoint that occurrence and hopefully intervene at that point. So we're going to go back and we're going to look here as early as we can. I'm not the only person doing this, of course, but I'm the one telling the story today. So how can we figure out where tau pathology first appears? Detectives, any ideas? How would you figure out where it is? Imaging. What kind of imaging?
Some molecular imaging with PET.
Okay, yeah. I think that's a great idea. He said PET imaging, looking for tau in the brain. I'm a little concerned that by the time you can see it on a PET scan, there's already quite a lot. So I want an even more specific way to look for the earliest signs of tau pathology. Any other suggestions? Yes.
You had said that some people, healthy people, have A beta early.
Yes.
Then you find the people that have high levels of A beta at a younger age, and they became candidates for tau later.
That is an excellent idea. And some people do that. Yes. So finding people who are either they know they have A beta, or another way to do this is they have high genetic risk, like it runs in the family, or they have genetic risk markers, and you look in them when they're 40 and try to see what's happening in their brains. There are whole communities that have high propensity for Alzheimer's. Like there's a group in South America that's being followed for exactly these reasons. So that's a great idea. Yes.
Markers in the blood that imply some increased chemical activity in the brain?
Yes. Maybe. So yes, looking for markers in the blood is a good idea. And this has a similar concern with imaging, which is that by the time you can see it in the bloodstream, which is quite separate from the cerebrospinal fluid and the molecular context in the brain, then maybe things have gone too far already. But there are blood tests being developed for phospho-tau. It's a big area of discovery right now. Yes.
The other markers,
Mm-hmm.
kinds of things.
Yes. So Alzheimer's also has a loss of smell identification early on. But the reason that for the loss in smell identification early on in Alzheimer's disease is because there's been damage to the part of the cortex that handles that function. So if it's already damaging the cortex, it's already spreading in the brain. We want to get there before that. I don't expect anybody to have this perfect answer, but I'm really glad you're thinking about it so well, because now I'm going to tell you the experiment that I think gave us the answer to this question. So it has 6 panels, and I'm going to uncover them one by one. This was a study done about 15 years ago, and it looked at cadaver brains of people across the whole lifespan, all the way from infants to very, very elderly people who had died just of tragic accidents, essentially. And the authors of this study examined their brains and they did a stain for phospho-tau, meaning they just looked everywhere in the brain, where are we seeing this type of bad tau accumulation? And they categorized their cases into cases with absolutely no tau, phospho-tau at all, and then in stages 1 through 6. And also there's a pre-stage 1 version as well. So when they look at the distribution of ages of people who have absolutely no phospho-tau in their brains whatsoever, this is the histogram of how old those people were. And they are basically little children. So when you have no phospho-tau, you are very young. Maybe there's like a 30-year-old in here, like 2. Pretty much just kids. This is stage A through C, which is the pre-stage 1, meaning these people had only the tiniest amounts of tau, but it was present in just tiny amounts. And they were in their 20s and 30s predominantly. Stage 1, these people are 40 to 50 with some, tails on either end. Stage 1 to 2, they're more 60 to 70. Stage 3 to 4, we're getting into the oldest old, 80, 90, 100-year-old. And stage 5 to 6, again, very, very elderly people in this group. So what we want to do is focus on this group, the people who had the tiniest amount of tau, phospho-tau, just in a few spots. And the insight here is that when you look in their brains and you ask, where was it? Where is the phospho-tau? It's always in one place. It's in the locus coeruleus. That's that crazy word that is difficult to pronounce. I still have to spell it coeruleus in my own head when I type it 1,000 times. That's where it was. That's where the phospho-tau began in pretty much everybody. That's why I study this region. So where is it? It's in the brainstem. It has nothing to do with the cortex, actually. This shows where it is. This is an image that that I made with the help of Dr. Voss here from the MRI facility that shows where it is. So the brainstem, this is just to orient you, this is the cerebellum, which is that little mini brain thing at the back of your head. And so this is the front of the head, and the brainstem is the part that goes down your neck. So what is this place? What does it do? It's called locus coeruleus, which literally means blue spot. And it is the blue spot because it contains neuromelanin, which is a pigment very similar to the melanin pigment in skin, just neuro because it's in the brain. And it looks bluish in brain slices. You can kind of see here, it's kind of a dark bluish color when you look at brain slices. There it sits in the brainstem, 2 little spots on either side. It has not that many cells, it's really quite small. What is it for? It's a neuromodulatory nucleus. So your brain works on neurotransmitters. Everybody's probably heard of that. They are the chemicals that keep your neurons talking to each other day in and day out and keep your brain working. But neuromodulators are a different type of neurotransmitter. They don't do the bread and butter every day, let's keep talking transmission. They do kind of a special type of transmission where they send big signals out to the whole brain at once or to large portions of the brain at once that allow the brain to move from one state to another state. The most famous one is dopamine. I think you've probably all heard of that. It delivers big changes in learning and motivation and reward. But the locus coeruleus makes one that is no less important, which is called norepinephrine or noradrenaline. They have 2 names for it. I don't know why we still use both of them, but we do. So it's a neuromodulatory nucleus. It makes the neuromodulator, this neurotransmitter called norepinephrine or noradrenaline, and it pumps it out to the whole brain. So this is where it sits in the brainstem, and all these red lines are where it sends noradrenaline. It is the only place that produces this in the brain, aside from a couple of spare extra cells. So it basically is doing 100% of the brain's production of noradrenaline. That's its job. What does noradrenaline do? What's it for? It is for modulating sleep and arousal. This region of the brain is the thing that activates to get you to transition from being asleep to being awake. And once you are awake, it takes you from alert to panicking and back again. So it modulates that level of arousal. Here's 11 having panic attack. It works on attention control. So when you're minding your business, doing your task, and something crazy happens outside and you hear a noise and you go, huh? That's the action of the locus coeruleus and norepinephrine, drawing your attention to something that might be relevant for you that you might need to pay attention to. It is super important for memory and learning. It allows a connection between what's capturing your attention and what's important and the memories you form. And it modulates the stress response, very similar to how the adrenaline in your body might work. So kind of a fight or flight modulation in the brain, like this freaked out ferret. Poor little fella. Okay, so that's what it does. That's what it's for. If the LC is important for Alzheimer's disease, what predictions about it would a good detective make? I want to hear your thoughts. If it matters, and all the stuff I just showed you about what it does, keep that in mind. What would we expect if it matters for Alzheimer's disease? Are there any predictions we can make?
Yeah, like behavioral problems, sleep problems.
Sure, exactly. Yeah, we would expect that whatever its jobs are, those functions will get worse. And they ought to get worse early in Alzheimer's disease, not later. If somebody has a problem when they're in stage 5 Alzheimer's disease, it's very hard to be sure about whether it has anything to do with the origins of disease. So yeah, do sleep problems have something to do with Alzheimer's? Does anybody know? Yeah, they do, for sure. Anxiety, how about that? Does that have anything to do with Alzheimer's disease? Yeah, especially early on, right, in prodrome. How about agitation, modulation of arousal? Yep. Memory problems, obviously. And apathy, which is when the locus coeruleus isn't working well enough and you're not alert enough and you're not able to kind of be attentive enough for the moment. Those things are all involved. We would also predict that people who have LC disruptions due to a different disease, like whatever else might mess with it, would be more likely to get Alzheimer's or less likely to get Alzheimer's, depending on what happened to the locus coeruleus. So PTSD is one of those things. So people with PTSD have measurable structural change in the locus coeruleus, probably as a result of being hyper-aroused all the time, being unable to kind of calm down. And PTSD is a risk factor for Alzheimer's disease. And another one is depression. Depression is also associated with measurable functional change in the locus coeruleus, but it's in the opposite direction of PTSD. There's less intensity when we look at that region with an imaging technique I'll describe in a minute, and it seems to be less active. Both of those groups are more likely to get Alzheimer's disease. Does that make sense? I'm genuinely not sure. I'm not going to present a tidy story here, but they're both involved with the locus coeruleus and they're both involved with Alzheimer's disease. Parkinson's disease I want to mention because although most people have heard that there's a very particular part in the brain that degenerates when Parkinson's disease happens, you may not know that the locus coeruleus actually always degenerates first, even in Parkinson's disease. First, we lose the locus coeruleus, and then the substantia nigra, the place that controls motor activity, that it makes that characteristic Parkinson's phenotype. And we would also hopefully predict that if a treatment has ever been found to change how the locus coeruleus works, it might also change Alzheimer's risk, one way or the other. Vagus nerve stimulation, which definitely changes how the locus coeruleus works, I'm going to talk about later. You don't need to know what it is now, but we're working on that one in our group. And selective norepinephrine reuptake inhibitors, which is a drug class that acts on the locus coeruleus and its modulatory neurotransmitter output, might also be expected to change Alzheimer's outcomes. And in fact, there are several clinical trials going on with these drugs for Alzheimer's prevention right now because of this. Okay, so I know this is kind of dry, but this is the mindset I want to be in when thinking about, does it really make sense that damage to this region is a really important key part of disease pathology? So now I'm going to talk to you about the work that I've been doing in my group to actually follow up on these predictions, to measure these things, to try to understand how we can identify early changes in the locus coeruleus that might tell us when somebody's heading down that path before it's too late to apply a disease-modifying treatment. So the first thing we do is we measure the locus coeruleus by basically just taking a picture of it. Until 2009-ish, this was only possible in cadaver brains because the locus coeruleus is tiny, really small. It's about maybe 3 by 10 millimeters long. And you definitely can't see it in any sort of standard MRI picture. But we now have special sequences, which I use with the help of Dr. Voss here, to take pictures of what it actually looks like inside a living person. And they're not very exciting to look at firsthand. This is an actual raw image. You can see that this fuzzy speck here, this kind of white dot here, that's the locus coeruleus. You can see one on each side. And we essentially go by hand and take a little pen and draw around the edges of it, and then we measure how bright the pixels are inside. So we measure how many pixels and how bright, and we also make things like maps like this that show where it's located in people of all ages. So what do we find by trying to measure this? Oh, a question? Okay. So we find that something changes in the locus coeruleus around age 60. And in fact, there's change before then too. So when we measure how bright the locus coeruleus is, starting with 18-year-olds, the youngest folks that are in our study, we see that they have a low brightness compared to those across the adult lifespan. Somewhere around age 60 seems to be the peak. The locus coeruleus becomes more and more intense. Easier to find on the scan until around 60, and then the brightness decreases. And this is the same kind of data. There's age on the bottom, and this is how intense the image is. And it's just a continuous curve that you can kind of use to estimate where the peak is. It's right around 59.5 years. So does anybody know why it would go up in the first place? That's it. That may not be unrelated, incidentally. The cause of this contrast, and by contrast, I mean very technically, like what causes the image to look brighter, is under debate right now. It's an area of scholarly interest, but we think it has something to do with neuromelanin. Now, neuromelanin, I already said, is the pigment that makes it bluish, right? And a funny thing I didn't tell you is that babies are born with none. They don't have any at birth. And it only builds up in there because it's a byproduct of the production of norepinephrine. So as this region works to do its job and pump out neurochemical, it also makes a polymerized waste product that's blue. So as a person gets older, you certainly see the accumulation of this signal, which we think is related to neuromelanin, although making a one-to-one correspondence turns out to be a little harder than we would all wish. So that explains why it's going up. But what explains why it's going down? Unfortunately, we think that's the death of cells, most likely. From cadaver studies that took MRIs and also just looked in the actual brains, as best we can tell, when the signal goes down, that's because the neurons aren't there anymore. Now, these people in this study were all healthy adults, so none of them had a diagnosis of Alzheimer's. Actually, that's not true. I had one person with an early Alzheimer's diagnosis, but she's not in this data set, and everyone else was a healthy adult. We tested their cognition. A few people had flags for maybe you should talk to your doctor just to make sure, but nobody was scoring in a high impairment zone. And yet we see this decrease. So that tells us that this region that we think is the origin of that tau pathology that later spreads to the whole brain is showing very clear changes starting around age 60, or possibly before, depending on who you are. So that tells us this is a good place to look. What else do we do to measure what this region is doing? It's nice if we can see if the cells are dying, but we'd really rather find out what they're doing before they're dying. right? When they're not dead and still savable. So we also measure their function. We do functional MRI, which this is the famous fMRI. The term means a lot of different things, but in this case, one way we can look at this region is to give people a task to do while they're lying in our MRI scanner. And this is actually not quite like any of the tasks I give, but it's one that I can explain easily. So for example, we give them an instruction like push the button when you see the big purple circle. And they're going to be laying in there, watching a screen, seeing circles flick by. Some of them are little purple circles. Some of them are little orange circles. They do nothing. And every once in a while, maybe 20% of the time, they see a big purple circle and they're supposed to push a button. And why do you think we give them that task? Any ideas? Exactly. Yep. It demands the function that the locus coeruleus is in charge of, and especially because detecting that this is what's called an oddball, a rare event that we need to pay attention to. That is kind of it's like a super activator for the locus coeruleus. We really need it to draw our attention to something that we're supposed to respond to. So we expect that when we give people a task like this to do, and we put them in the scanner, we measure what their brain is doing over time while they do the task, we ought to be able to see if the locus coeruleus is responding in an expected way. Now, the details of making this work turn out to be rather complicated because, as I mentioned, it's a tiny brain region, and functional MRI traditionally doesn't do well with deep brain structures that are like way in there, and also when they're tiny, and also when they're right next to places that are not brain, like your ventricles, which are full of fluid, and then you have to breathe in and breathe out, and the fluid moves around, and it's actually quite complicated. But with enough technical fussing, we can get there. So what do we see? So when we look at performance on a task like this, we can look at a graph like this one. So on the bottom is time. And here is, you can think of it as how much activity in the locus coeruleus went up. So did we get a response or not? And you can expect that in this type of graph, we're only talking about one event. So you can just imagine a person sitting there, nothing's happening, we're waiting, and then boom, something happens on the screen. What did the locus coeruleus, this mighty little guy, do? In young people, when a target event comes on the screen, that's the red line, you get a big response. Locus coeruleus is like, whoa, I saw the thing. And the person activates their systems that they need to push a button. And when a distractor appears on the screen, that's the orange line, this is something that's almost like a target, but not quite. You're not supposed to push the button. We see a response, but it's not very big. And when nothing much happens, a blue line, it doesn't do much. Even if there were pictures or something else interesting happening, if you weren't waiting for that thing that you were told to push a button for, you're not going to see a big response. In the middle-aged people, which in this study is about 45 to 65 years old, what do we see? Yeah, maybe a little bit more distractor responding, a little bit like hot to the button, maybe a little bit, but kind of the same. It's not too different. And then in the older group, what do we see? Yeah, there's greatly reduced responding in our older group. They do have the same kinds of patterns. It's hard to see here, but if you kind of zoom in on this one, you can see that there is a little bump for the target, and it is a little bit bigger, but it just isn't nearly as strong as what the other groups are doing. So when that big purple circle shows up and the locus coeruleus is like, whoa, I saw the target, pay attention, push your button, that response seems to diminish in older age. And in middle age, we see some changes. We see what looks like a little bit of hyperactivity, a little bit of like too eager to respond, maybe. Here's another way we try to measure this tiny little tricky brain region. We try to take a shortcut. MRI is expensive and difficult, and analyzing it is hard. So what else can we do? We can use pupillometry. Why pupillometry? What even is that? So pupillometry is measuring how big the pupil is in the eye. And this would seem, quite reasonably, to have nothing to do with the locus coeruleus, but by a quirk of evolution, it does. So the region there's a connecting region between the locus coeruleus in the brainstem and the muscles that control your pupil dilating muscle. And they're actually very closely connected. So when your locus coeruleus fires a lot, your pupil tends to dilate more. And when it's not firing very much, your pupil constricts. This is probably related to some evolutionary function, like if there's a dangerous thing happening or a sudden noise, you want to take in more information, all of a sudden look around, get your bearings. But these days, it happens even when a target appears on a screen and some tiny little micro surprise happens to us. We still see that dilation. So we can try to use it. In our lab, we take pictures, videos of the pupil. You can see, if you watch, that it's getting larger and smaller. And we essentially just measure the diameter. And like I showed you with the other task, we measure what happened right after something interesting came onto the screen and see what kinds of responses we get. So this is a complex figure, but I'm just going to draw your attention to the green line here first. The bottom axis is time. So this is how big the pupil is, and this is over time. And it's about 2 seconds. So we're talking about pretty quick responses. So when something happens on the screen that's a target, like the purple circle, you get whoop, the pupil gets bigger over the course of about half a second, and it's going to go down by about 3 seconds. And in the middle-aged adults, it gets actually the biggest of all. We have the largest pupil responses. And in the older adults, the response is getting smaller again. So this is telling us another piece of the same story. There's reduced responding of systems that are linked to the locus coeruleus in older adults. And in the middle-aged adults, we see something like hyperactivity. And I wanted to draw your attention to this. You remember this figure from earlier? There was this extra blue line, which is a suggestion by some researchers that there's a hyperactivity phase that happens early on in the Alzheimer's disease process. And they didn't necessarily mean pupil dilation when they made this figure. They might have been thinking about especially glucose metabolism, which is a big deal in the brain and could have to do with the onset of that inflammation that happens in the brain. But it may apply to other systems as well. Other cells that are stressed, other cells that are trying to work harder and do their jobs kind of extra, even though they're experiencing some damage. So what are we doing about this so far? We have some pretty convincing evidence from lots and lots of researchers, and even in our own hands, that there is functional change in the locus coeruleus, certainly in older age, and also in midlife. Can we do anything about it? One thing that we're trying is vagus nerve stimulation. So the vagus nerve, anybody heard of it? Probably most people have, right? It's kind of everywhere. It does a lot of things. It seems to have so many jobs that it's involved in everything. The word vagus comes from the same root as vagabond, and it actually means the wandering nerve because it's everywhere. It does the whole torso. It carries all that information up to the brain. And it famously innervates the heart and helps you control your heart rate. It does a lot of jobs. It innervates the gut. I think that's probably where most people have heard about it lately. But when it comes up to the brain, especially up on the left side, it stops. The first place it stops when it comes up into the brain is the nucleus tractus solitarius, which controls heart rate and breathing. And then it goes straight to the locus coeruleus. So that's basically stop number 2 on the vagus nerve route to the brain. And because of that, if we send signals up or do something to the vagus nerve, we may be able to change activity in the locus coeruleus. And another further convenience for the researcher is that there is actually a little piece of the vagus nerve that innervates your outer ear. And that's great because the vagus nerve that many people actually use for treatment is buried in here. It's like in your neck, and you have to have a surgery and stick a little electric thing in there and wrap it around your nerve and then implant batteries, and then you can never have an MRI again, or only under the right circumstances. But we don't have to do that. We can just stick a little buzzer on your neck or on your ear and go through your skin. And lots of people have done this. We're certainly not the only ones. And it definitely has effects. It is now FDA approved to treat 2 conditions, depression and migraines. And it's being evaluated for several more. But one of the most interesting things about it is that it was actually discovered to have a cognitive and mood effect by accident. Does anybody know what condition vagus nerve stimulators were first built to treat? It's actually intractable epilepsy. So people who had failed dozens of seizure drugs and were still having seizures, and they really needed seizure control. And they have implantable vagus nerve stimulators put in and turned on. And some of them get seizure control and some of them don't. But researchers noticed pretty quickly that people, even who didn't have any improvement in their seizures, had better mood and better cognition. I thought, huh, so this is doing something in the brain besides calming seizures. So we're doing this now. We're doing vagus nerve stimulation while people do a task. And we're doing real stimulation and sham stimulation. And the difference between the two is just a slight different position in where the stimulator goes on the ear. Because in one position, it gets that branch of the vagus nerve. And the other one, it just buzzes your ear. So in these graphs, I'm going to use pupil AUC, which is a measure of, if you think about the graphs that I showed you before, where there's time on the bottom axis and pupil size here, AUC is a measure of how much stuff was under this curve. So in younger adults, when we deliver, we have them do a task with sham and real vagus nerve stimulation, we see that when oddball events, target events, come up on the screen, their responses, comparing these two blue circles, get a little bit bigger. So it's significant. There is a little bit of a boost. And these constrictions, which are actually to bright stimuli that pop on the screen, get a little bit larger in the negative direction. So interesting. What happens in older adults? Anybody want to say what they see happening in the other age group here? Yeah. So it's an opposite effect. Instead of increasing pupillary responses while people are doing this task, the delivery of real but not sham vagus nerve stimulation seems to suppress these responses. Why would that be? Vagus nerve stimulation seems to do something different in older adults. And this is a really interesting question for me. These are the hottest new results that just came off the press 2 weeks ago. And this is where I'm really thinking about, what does this mean and what does it mean for the future of this research? We already know that there are changes in middle age and in old age. We know that the LC is working differently. It's a little bit confusing, but we think that the locus coeruleus may be working harder at baseline, trying really hard, pushing out a lot of norepinephrine to try to keep up with the brain's need, to the point that it doesn't have any extra to do phasic responding. Phasic responding is what we call those little blips that we get when something exciting happens. So in other words, it's putting all its effort into keeping you like alert and focused. And there's nothing left for like sudden reorienting of attention in the moment on that kind of fast basis. These are two other ways to look at this phenomenon. This graph here shows how big target responses were in sham or real vagus nerve stimulation. And the gold dots are older people, and the lines generally go down, meaning their responses got smaller. And in younger people, the lines generally go up. There's a couple of people for whom that's not true, these two down here. I don't know what's up with them. And here we look at the baseline pupil size. So this isn't phasic responding. This isn't something cool happened on the screen. What happened to the pupil? This is just how big was the pupil generally the whole time. And in younger people, the application of VNS doesn't seem to make any difference so far. This is pilot data. But in older people, it's notably different. We get a larger baseline. Yes. In this particular set of data, because it's pilot, this is a split older than 50 and younger than 50. In the other groups, it was 3 groups, 18 to 45, 45 to 65, and 65 and up. So let's pause. We know that the locus coeruleus is the first site of tau pathology, and this appears in young adulthood. We know that the LC experiences measurable functional and structural change in mid and later life. And if we get good at understanding these, we might be able to pinpoint these problems before the whole brain is affected. We think we're able to affect this function with vagus nerve stimulation, and it seems to work differently in younger and older people, which, as a side note, could make it useful as a diagnostic. And we know also that most people don't get Alzheimer's disease, even though everyone gets tau pathology. So what can we make of this? I'm going to go through these quickly because we're going to run out of time. But the thing that I think is really important here is that there are many modifiable risk factors for Alzheimer's disease that make it so that getting tau pathology doesn't mean you get disease. Getting amyloid pathology doesn't mean you get disease. And I want to know, do these things relate to the locus coeruleus? Do they have anything to do with this system that we think is this most vulnerable early place? Can we steer off the road? Probably. A lot of latest research is saying, oh, it's preventable. Lifestyle interventions, you can measure how much is preventable. The estimates range from 40% at a minimum to 90% in some people's estimation of how much Alzheimer's is preventable. So I think that's very worth talking about. The locus coeruleus is tiny and it has a huge tree that goes to the whole entire brain. That makes it a vulnerable place for toxins. It's also located in a place in the brain that gets a lot of exposure to the fluids. So some people feel that avoiding toxins, which means avoiding contact with a football field size amount of area in your locus coeruleus neuronal tree, is a great way to keep it healthier. Exercise has been shown to be directly related to how the locus coeruleus functions. This is how bright it is on an MRI scan, and this is how good your aerobic fitness is. There's a relationship there. And we know that exercise improves our Alzheimer's odds. Diet quality, there's so much work here. I'm not even going to say much about it because we're going to run out of time. And social connectedness, which is a really big deal for maintaining your alertness and arousal levels in a way that doesn't push you into hyperarousal and PTSD and the kinds of conditions that we know increase norepinephrine release constantly, build up that norepinephrine and may challenge this system beyond its limits. And again, this has been already directly related to the health of the locus coeruleus in the research. And last, sleep. Locus coeruleus is an architect of sleep-wake transitions, and it must be off for REM sleep to proceed as it should. There are researchers that have their whole career based around this system, and it's really amazing the work they've done. And we know that in Alzheimer's disease, it's not as quiescent in sleep as it should be, probably because of that hyperactivity. So that's another place to look for locus coeruleus connections. So we're coming to the end here. This work involved 213 participants who came and joined us here on campus, and quite a number of RAs, as well as my mentors and collaborators. Thank you for your time.
Thank you so much. We're going to move into our short Q&A portion. Does anybody in the room have a question for Dr. Riley?
For animal models, with animals, one can do much more experimentation than with humans. And I think there are these Alzheimer's mice. Do you know anything about it? If we
Yeah, so there are some
get
amazing labs working on animal models in Alzheimer's disease, and they've come up with some fascinating conclusions that we can't get in humans. One of the ones that really is on my brain at the moment is that if you overexpress neuromelanin in mice, they don't naturally produce it, by the way. They make their norepinephrine, but they don't make neuromelanin. I don't even know why. But if you make it so they do, that neuromelanin builds up and builds up and builds up, and they're doing fine. And then all of a sudden, boom, they get really sick and the cells all die. So we think that having too much neuromelanin is actually a player in this disease process. Another thing you can do in animal models is look at whether tau in the locus coeruleus literally spreads along its axons up into the brain. And there is evidence that it does. So if it appears there first, it may literally be the kind of typhoid Mary spreading it to the rest of the brain. It's early work on those.
My question was maybe a little out of scope, but I was just, some of the early graphs you were showing, it made me wonder, if you could somehow prevent A beta buildup, would that prevent the tau buildup later?
That's the perfect question. I think A beta doesn't seem to have a clear starting point the way tau does. And it's a much less specific marker. So there are people that have brains full of A beta and they go on to live long, healthy lives with no Alzheimer's disease. But it is a predisposing factor. And the more you have of it, the worse the odds, even if it's never a great predictor. So I think now that we have this better model of kind of the multiple hit hypothesis of how disease develops, understanding the interaction between the development of A beta and the development of tau is going to be a next frontier, trying to figure out why do they have this kind of synergy together? Why does one predispose the other? And I don't know the answer.
Thank you so much for this talk. I don't know that we have awards for best talks, but this would be high among them. So I was sort of enamored where you ended, and I know we've run out of time, but you're kind of going through potential interventions or things we could do. Yeah. And I think as you talked about the arc and the historical arc of arriving here from sin to weather to now saying, well, we actually think we know where in the brain this is sort of starting. The turning point for me was when you talked about the vagus nerve and the practices and behaviors we're able to do to interact with the brain. And you mentioned the gut and things, but the way I was introduced to the vagus nerve is more through maybe blood pressure or breathing almost.
Yes.
Is breathing implicated in defending against development of Alzheimer's if you are regulating?
Yes. In fact, there was a study a year ago by one of the people I consider a hero of this field, Mara Mather's group in California, that asked young people to do biofeedback deep breathing practice. And the biofeedback was to increase their heart rate variability. So they were trained on the type of slow breathing that's like we know is the most beneficial. And they actually reduced their locus coeruleus intensity on neuromelanin scans. So clear structural change in young people. And it was only 5 weeks. So yes, I would say there is a clear implication. This probably is also the reason why meditation training is sometimes linked with reduced cognitive decline. Although that link is tenuous over a lifespan and over all the conditions that we all face. And then there's this argument about whether we want an optimal amount of stress, not too little or too much. So I wouldn't say we've kind of nailed that down, but I think there's definitely connection. Oh, I see. You've got the mic. Sorry, go ahead.
Thank you for the talk. And I want to ask, like, we all know that some genetic factors also play a role in AD.
Yeah.
So are there any genes associated with the function or structure of this blue spot? Like,
I don't know of any. With the big risk factors in AD are, the ApoE gene, which is a cholesterol gene. And the question of whether that affects tau buildup, the way it affects amyloid buildup, I don't know.
but any genes that encoded for this blue spot structure?
So I don't know about structure, but we are working on a candidate in our group. We're working on the ADRA2B gene, which is a adrenergic receptor. It's actually an autoreceptor, so it's the type of receptor which ought to help the cells self-regulate how much norepinephrine they're producing. And there are 2 or 3 reports so far, and we're hoping to be another one if we can get our PCR to work, that having a variant of this gene that likely results in more norepinephrine release is associated with reduced cognitive decline. So it's a great prediction, and it is something we're working on, and there are signs that yes, that is the case.
Thank you.
Mm-hmm.
We'll take these 2 questions from online and then we'll wrap up.
All right, so the first question is, do you use some generic vagus nerve stimulator or one your lab produced for this purpose? Signed, a migraine sufferer?
Sufferer, yeah. We have a research device, and we're supposed to get a second one that was supposed to arrive last week that hasn't arrived. The Gamma Core device is the commercially approved device for migraines. We are not using that device. We basically have something from a little German company called TVNS Technologies, but it's really simple. It's basically a little metal thing that you clip on your ear and it just delivers a pulse. It's essentially a TENS machine. And this machine that we're going to work on with Dr. Voss evaluating the efficacy of this treatment inside the scanner is a commercial TENS machine. So it's a big neuromuscular signal generator. And you just attach wires and little stickers on the end, stick it on a person and send some electricity through it. So it isn't the gammaCore device.
Okay. And then the next question is neuromelanin sounds like a sinister background actor here. Is there any indication that finding a drug that could help the LC clear neuromelanin could forestall degeneration? Is anyone working on that?
Okay. I agree. It sounds like a sinister backstory, but it also has a heroic story that I didn't really mention, which is that it's thought to sequester toxins. It's actually thought to be a sponge. So it gets actively stored in these little granules that are hanging out in your big neuromelanin cell or big norepinephrine cells, they're actually really big. They're like bigger than normal, kind of fat, chubby cells with lots of water in them. And they have these little pockets that neuromelanin accumulates in, and they actually soak up oxidative radicals and probably pollution and toxins, and they store them in there. So it's a difficult balance. There's papers that call it like a riddle and an oxymoron, and there's so many people that are confused about this, including me. You don't want to have too little because then you can't soak up toxins, but it seems like there's a tipping point. And when you get too much, it's not good for you. So yeah, I think maybe at some point, maybe the problem is you stop being able to clear it. And the study from Mara Mather's group last year suggested that deep breathing can help you clear it, even in young people, which we used to think that was impossible, that we only accumulated ever up until the cells died. Now we know that's not true. We have to evaluate whether we can improve clearance. As for drugs that do that, I haven't heard of any. Maybe just deep breathing for now.
We give one more round of applause to Dr. Riley.
Yeah.
Thank you so much.